Hi everyone. In this video, I'd like to talk a little more about our course and go over the main parts of the DNA barcoding process. So what is DNA barcoding? DNA barcoding is a method of species identification using a short fragment of DNA, which we call a DNA barcode. You can think of the DNA barcodes as the unique species identifiers, which you need to scan in order to identify the species. This works similar to scanning the universal product codes in a grocery store. Each product has a barcode, and at checkout, barcode scanners will read the bars and identify the code of your product. There are multiple sources of DNA: species tissues, species gut contents, or species feces. These are just a few examples. So how do we do DNA barcoding? First, we use a small piece of tissue from the species we are working with, and we extract genomic DNA. Next, we target a specific fragment of DNA, which we use as a DNA barcode, and we amplify it using a polymerase chain reaction, or PCR. Then often we want to confirm the presence of the amplified DNA fragment in the PCR product. To do this, we use gel electrophoresis to visualize DNA. Finally, we obtain a DNA sequence for the DNA fragment, which we isolated. DNA sequencing is typically the final step of the DNA barcoding process, but it is not the final step of the research project, of course. Now that you have DNA sequences, you can do many interesting things. You can identify species, you can determine the species diet, or construct phylogenetic relationships. These are just a few examples of what you can do. In our lab, we do a lot of DNA barcoding work to identify plant-insect interactions. Here you can see what our work looks like. Typically, we collect insects and plants. We extract DNA separately from the plant samples and from the insect gut contents. Once we obtain the sequences, we identify the ingested plant species. Having this information, we can conclude what plant species have been consumed and whether the insects consume the plants from which they were collected. Over the past several years, we developed the protocols for each DNA barcoding step. It's very important to follow these protocols to make sure you always add the right amount of the right reagent. Here you can see the lab where we conduct our DNA barcoding work. As you can see, each step of the DNA barcoding work has its designated place. It's very important to separate DNA extraction, PCR, and gel in space, as this prevents cross-contamination of the samples we work with. Here you can see our DNA extraction station, which has all the supplies needed for DNA extraction. Our PCR station is in the middle of the lab. Here we do PCR, DNA purification, and all the preparations for sequencing. The gel station has its own space where we keep all the materials and supplies we need to run a gel. Finally, once we finish DNA purification, we can send the samples for sequencing. For sequencing, we use a sequencing company, which is called Azenta, former GeneWiz. We have a Dropbox, which is conveniently located on the second floor of our plant sciences building. Depending on the sequencing service we use, we can receive the sequences back either on the next day or in about a couple of weeks. That's it. I hope you will enjoy our DNA barcoding course, and I encourage you to review some additional resources too. Here you can see just a few of them. You can also check our papers to learn more about the projects which we are doing in the LAMP lab. Thank you and happy DNA barcoding.